Thank you so much, Oliver. And thank you all for joining us this morning for this inaugural Stanford Gynecologic Oncology Research Seminar in honor of Dr. Berek. I am delighted to serve as the moderator for our first session, really focused on genetic and molecular alterations in breast and gynecologic cancers. And I'm delighted to honor and welcome our keynote speaker, Dr. Joellen Schildkraut. Dr. Schildkraut is a professor in the Department of Epidemiology at the Rollins School of Public Health and is a member of the Cancer Prevention and Control Research Program at the Winship Cancer Institute of Emory University in Georgia. She received both her MPH and her PhD from Yale University, where her doctoral dissertation and early publications were focused on the genetic overlap between breast and ovarian cancer through the examination of family history. This work contributed to the discovery of BRCA1 and BRCA2, as we all know, two very highly penetrant genes associated with increased risk of breast and ovarian cancers. Dr. Schildkraut has authored over 360 peer-reviewed journal articles, and in addition has contributed to numerous book chapters and textbooks. Please join me in welcoming Dr. Joellen Schildkraut. Thank you.
I am not exaggerating. I'm so honored to be here, especially for Dr. Berek. I'm so impressed with his illustrious career. And I now know there are other things I'd like to discuss with you, research related, but because I learned that just now. But I thank you so much for inviting me here today. And I hope I could do some justice to genetic epidemiology of breast and ovarian cancers, because they really come together. So I just, okay. So I just want to make sure I know how to advance. I'll just do it this way. Okay, so this, my talk today is going to go through decades of my work, as well as reflecting the research done in the area of genetic epidemiology of these cancers, both breast and ovarian cancer, because they go hand in hand. Starting in the early 80s, actually, while I was doing my PhD work, we were getting the description of familial breast and ovarian cancer was being published either in pedigrees or in case control study, which is something that I did. And then as we go through the decades, and starting in 1990, this is when BRCA, I'm sorry, I say BRCA 1 and 2 because I'm old, not BRCA, but I'll try, I can't switch easily, so I'll just say BRCA 1 and 2. I was in Research Triangle Park, right where some BRCA 1 was being positionally cloned. And I was interacting with some of the people that were involved with that, as I was also doing some work in that area. So it was an extremely exciting time and extremely competitive and time to find those genes. And I did a little bit of contribution. I don't know how much, but I contributed some to that area. And then as time went on, we got into the era of GWAS, genome-wide association studies. I will say, looking back, I'm a little disappointed. I don't think it was the breakthrough data that we were hoping to generate. I think we learned some, but that went on for a while and took up a lot of time and money from NIH, and I was part of that as well. And then to present time from my career, I've been focused on the molecular characterization of ovarian cancer, as well as the epidemiology. I do the full, look at risk factors, look at prognostic factors, and look at the disease itself in terms of the description of the tumors. So I'll just take you really quickly. This slide represents why this work has been so challenging. When we focus on ovarian cancer, I call it the perfect storm. It is rare, it is highly fatal, and it is heterogeneous. And for a genetic epidemiologist or epidemiologist, that just presents lots of challenges. And when I'll try to even point this out as I go through some of my slides. So ovarian cancer is rare. There are approximately 22,000 or so women diagnosed each year with ovarian cancer, and it makes up about 3% of all new cancer cases in women. And it's about 5% of the deaths. So in this slide, you could see that if you were diagnosed with early stage disease, you would have a very good survival rate over 5 years, 93%. But as you get further down into the stages of diagnosis, it gets pretty depressing. So if you're at late stage, it's 30% or worse, 5-year survival. And so the majority of women are diagnosed at late stage. This is epithelial ovarian cancer. And there's no effective screening for ovarian cancer because the symptoms are vague. It is rare. There are a lot of challenges in developing a screening approach. This just gives you an idea of how the incidence to the death to incidence ratio for ovarian cancer and how high it is compared to other female cancers. And again, as I mentioned, epithelial ovarian cancer is a heterogeneous disease. There are some 5 major histotypes that are considered in epidemiologic studies and genetic studies, including the most common one, which is high-grade serous ovarian cancer, which does make up about 70% of all epithelial cancers, but it goes down precipitously after that with endometrioid and clear cell, approximately 10%, and mucinous cancers and low-grade serous cancers are even more rare at 2 to 3%. This slide just depicts that even when you stratify by stage, that how the histotypes differ in terms of survival. And I guess we could have a long discussion about that, but I'm not planning that now. But the best survival times are with low-grade serous and endometrioid, regardless of stage, and the worst is carcinosarcoma, which makes up about 5% of all epithelial ovarian cancers. So my talk is going to cover 3 major areas, including family history of breast and ovarian cancer and the breast ovarian cancer syndrome that led to BRCA1 and 2. And then I'm going to talk about the Ovarian Cancer Association Consortium that became a big part of what I was doing starting around 2005. And then into what my more current work is in terms of gene expression and subtypes of ovarian cancer. So it was the motivation to look at genetic factors in breast and ovarian cancer were the observations that were made in families, and that you could see in the pedigree there, it's showing transmission through 3 generations of breast or ovarian cancer. And these families were described. And then as well, the family history of ovarian and breast cancer were looked at in epidemiologic studies and reported where the relative risks increased with the number of affected family members and the number of early onset cases. And those were hints about that we were looking at a genetic syndrome. So the work that I did started with my dissertation, so it's so long ago, but it was I used the cancer and steroid hormone study, or was also called CASH, and it was focused on hormones, oral contraceptive use, but they did collect a lot of family history that became really generated a lot of information, and it gave us a lot of insight, not just the work I did, but others. But what this slide shows is when looking at the family history of ovarian cancer cases and controls, breast cancer cases and controls, and endometrial cancer cases and controls, that we're in the hundreds. This is not a huge sample of only 493 ovarian cancer cases, for example, and many more controls, but that were non-cancer women didn't have any of these cancers. The results suggested that a pattern, we can see it. We saw a relationship. If you had ovarian cancer, you had increased family history, higher relative risk of having a family history of ovarian cancer, as well as breast cancer, but not endometrial cancer. If you were diagnosed with breast cancer, you had a higher relative risk of having a family history of breast cancer, as well as ovarian cancer, but not endometrial cancer. And if you were diagnosed with endometrial cancer, you had a higher risk of a family history of endometrial cancer, but not necessarily ovarian or breast cancer. So that was confirming, or more data to support this breast ovarian syndrome. So as I kept on going, I was in my career, I was very inspired by the discovery of tumor suppressor genes for hereditary cancer. And this was proposed by Dr. Al Knudsen at the time, and his two-hit hypothesis for how tumors may arise from two mutational steps. His paper, I still keep a copy, and I keep a copy of his picture because this is really what I think it grabbed me so much. But in this example, it's retinoblastoma, where those with an family history had bilateral retinoblastoma, and those without family history tended, if they developed a retinoblastoma, they had unilateral. So the hereditary form was shown to arise from a single somatic event in a cell which carried a mutation. This is the hallmark of a tumor suppressor gene. So this is the RB gene in this case, but it's a model. And again, as I said, I was very inspired by his work. And it was a mathematical depiction of this two-step hypothesis for developing familial cancers. So what I did is I looked at multiple primary breast ovarian cancer cases. So it wasn't multiple primary breast or multiple primary ovary, but it was both, because this is what the syndrome was kind of cluing us into, that these two cancers were related. And at this time, Mary Claire King had done the linkage in 17q. So at this point, we knew something must be going on in chromosome 17q. So I assembled these sets. They were matched sets of tumors from the ovary, breast, and normal tissue. And I worked hard to do that. I had a little money from NIH to do it, but not a lot. And I obtained paraffin-embedded tumor blocks. This is my life has been with FFPE tumor, paraffin-embedded tumor tissue, because getting fresh frozen is very, very difficult, even though that would have been my first choice. But to look at this set of cancers, that was our option. It's been kind of the theme for a lot of the work that I've done. But I managed to assemble 24 multiple primary cancers of the breast and ovary. We extracted for the DNA from the tumor tissue, and I got to go in the lab and do a little bit of that work. But anyway, and then we also identified 4 polymorphic microsatellite markers. This is a long time ago. So we didn't have, the genome wasn't cloned yet. I mean, we just didn't know a lot at this point, and we didn't have the sequencing data available. So at this point, it's really in the dinosaur era, I suppose, of doing this kind of work. And what we were looking for were informative or heterozygous or wild type mutant cases where to look at allele loss. And allele loss was scored as 2 times the ratio of one allele to the other in cancer compared to the normal tissue. And in this case, we used audio radiogram. So it really does seem like a dinosaur. And this is what the data looked like. And we looked at the loss again. We would look at the paired tumor tissue, either in the ovary and breast, as well as normal. And we looked for allele loss and to pinpoint what might be going on. And this is actually before BRCA1 was sequenced. But I was doing this and it's a longer story I don't have time to tell, while other labs were approaching it in a different way. So the results of that study really came out after the sequencing of BRCA1, even though we were doing it simultaneously. It's just when finally things came out. But here, the major results were that the loss of heterozygosity at that area of BRCA1 17q was in 46% of breast cancer cases and 78% of ovarian cancer cases. And we also noticed that it was the same allele that was lost when it was lost. So we looking at these women with these tumors, we noted younger ages of onset. It was all consistent with a genetic syndrome. So the conclusions from this was that loss of heterozygosity from multiple primary tumors suggested that BRCA1, a gene that at this point we did know about BRCA1, confers susceptibility to ovarian cancer and early onset breast cancer, and that the region of loss was also seen in sporadic cancer cases that didn't have a family history. So we didn't always, it was not always seen, but we think that we were getting an idea that this was an important region, whether it was a familial genetic associated cancer or not. So that was the early part of my career. And then as time was going on, I became one of the founding members with Andy Burchuk of the Ovarian Cancer Association Consortium in 2005. We all met at Cambridge and had a lot of fun there. We went punting on the cam and all those things. But we did start forming this this consortium. We mapped out the type of data we wanted to collect and what the purpose of it was. And this consortium really became a lot of part of what I ended up doing, not only what I was doing, but it really did take up a lot of my time. And so the OCAC, which we refer to it as OCAC, we scoured the world to get cases and controls of ovarian cancer. It was focused on genetic association. And ovarian, remember I mentioned rare disease, heterogeneous, very challenging. And compared to other cancers, it is, like a breast cancer would be hundreds of thousands of cases. Here we were able to only put together about 23,000 and 40,000 controls. And for GWAS studies, you really need more than that. But at the same time, there were data being generated for genome-wide association studies. And these are where you look at a number of biallelic variants of SNPs that are genotype all across the genome, but they're not necessarily SNPs with any real biological meaning at all. They're just variants all the way through the genome. So you could try to see where maybe there might be a putative gene. And they're easy to genotype and relatively inexpensive these days to do this work. But the associations are relatively low. The odds ratios tend to go from like 1.03 to 1.29, or if it was protective, 0.74 to 0.9. That is in contrast to a major gene like BRCA1, which would at least be odds ratio of 1.5, but usually a lot more, a lot higher than that. So major genes, you get a stronger association. These are very low level, weak associations. But to date, there have been about 40 genomic regions that have been identified through this work. And again, I think that if you look at other cancers, you'll see a lot more. And we were challenged by all the attributes of epithelial ovarian cancer. And I think we reached our limit, close to that at this point. There are more recently rare variants were genotyped, and there are a few more added into that number. But all this work can be used to develop polygenic risk scores, and I will show you a little work on that that's been done. And that's the sum of all these SNPs that are associated, and they're weighted by the effect sizes from the GWAS studies to develop this score where you add those together. And it's a way to kind of stratify people into risk groups, high risk and low risk. So then it's more challenge to what I was doing. I decided to study this cancer in African-American women and established a case control study that a lot of the data in Black women is coming out of that. But ovarian cancer in African-American women or Black women is lower, it's lower in incidence. So it's about 2,000 per year in Black women compared to 18,000 or more in white women. So it's more rare. I mean, there are less Black women at risk, and has a lot to do with it. And the incidence is, as I said, lower, but the 5-year survival rate is worse. And so 20% of all ovarian cancer patients will not survive the first year of diagnosis based on data, but 30% of African-American patients die in the first year as a contrast. So I did allude to the fact that I created I was funded to study African-American women. It's the only study that I'm aware right now that was really focused on Black women. We call it ACEs, or the African-American Cancer Epidemiology Study, and Melissa's been part of this for a number of years, Dr. Bondi. So it's a multi-center study, population-based, women that self-identify as Black, and we were in lots of different regions in the country, up to, I think, 13. And after years of accruing patients, we now have 700 cases and 700 controls. We collected a lot of information on these cases, and you'll be seeing how we have parlayed that into some of the molecular studies that I'll be presenting. We did do some GWAS. in this group, along with the OCAC data that was assembled on Black women, but it was anemic, very small numbers where we only had, I mean, it's amazing. There are only 750 some odd cases of self-identified Black women and about 1,200 controls to compare to. And we did do the GWAS. We did compare our findings to that in whites, and most of the time, not all of the time, we did not see in our top hits that did not quite meet GWAS significance, but were kind of suggestive. We did not always see a relationship in whites. So there's some differences. There are possible explanations for that. But the top genes that we did find were in the high-grade serous that were definitely suggestive of a association. One was FST that encodes a gonadal protein and inhibits the release of folate stimulating hormone. And it's also been linked to PCOS or polycystic ovarian syndrome. Since we published that, the group in OCAC did look at it using more SNPs and rare SNPs and found an association with a much more rare variant in whites, not the same one. This has happened more than once. And actually the next gene on my list, which was the melanoma-associated antigen gene family, it turned out MAGCE1, we found that was our next highest association. And it didn't look like it was associated in whites, but that GWAS and rare SNPs did find something not the same SNP. And this SNP I was really interested in because it was another gene in this family was linked to familial cancers in ovary, and it's a possible target for immunotherapy. And then the other two, one was a long non-coding RNA gene, and the other one with maybe unknown function, and the other GK2 encodes glycerol kinase 2. So I mentioned polygenic risk scores. So this is what the goal right now is of calculating those. You can identify those with using hundreds of variants to identify people at high versus low risk and maybe target the high risk group. So more work was done in the OCAC using some of the data that I contributed, but I was not a major player here. But it was a doctoral student at the University of Cambridge who published this paper and was able to calculate PRS scores for white women, Asian women, Black women, and people that were carriers of BRCA1 or 2. And basically, what was interesting here was the PRS score using the same SNPs was very similar in Black and white women. It was lower among Asian women, but also the magnitude was not all that different among BRCA1 and 2 carriers. These scores do not include BRCA1 or 2. So it's just these variants that have been associated and actually many more variants. It could be like 1,000 or more variants that were found to be associated in GWAS studies. So now we have a way of trying to identify women, maybe that we could stratify the risk on women with in terms of developing ovarian cancer, but is there clinical utility to this? And so far unrealized, but there might be, but I don't think it's been used. So another area that I started, because of the frustration with the rarity of ovarian cancer, I came up with an idea in talking with colleagues, of course, on looking at pleiotropy of breast and ovarian cancer to try to leverage the larger size of breast cancer cases that we had access to with the more rare and smaller sample sizes of ovarian. So if we could find an overlap like we can with BRCA1 and 2 and other genes, maybe we could learn more about genetic susceptibility by looking at at that overlap. It keeps on coming up in my career, genetic overlap of these two cancers. So it was motivated by what we do observe with the high-risk cancers in BRCA1 and 2. There were already noted GWAS associations in breast and ovarian cancer that overlapped, especially correlations with ER-negative breast cancer and serous ovarian cancer. And there were some common variants in an analysis done to look at the pleiotropic effects. So this led me into a transcriptome-wide association studies you may have heard of, TWAS, to examine genetic pleiotropy of breast and ovarian cancers. And we just completed most of this work about a year or so ago. But generally the approach here is using GWAS and using, there's an available resource called GTEC, or the Genotype Tissue Expression Project, that basically it's cadaver samples with matched genetic information to look at the association with gene expression and genes. And then if you find them, you bring it back, that information back to a GWAS study to see if you see it in a GWAS study of ovarian cancer or breast cancer. So we use gene expression to predict for gene expression prediction models and GWAS data to improve the power. From this study, we used the OCAC data and a very similar consortium called BCAC that is focused on breast cancer, and the gene expression data from the GTEC. And from all that work, our first paper identified 14 candidate genes and for breast cancer and 8 candidate for ovarian cancer that were not in the areas of susceptibility from the GWAS study. So these looked like they were different. There were 38 candidate genes for breast cancer and 17 for ovarian cancer that were in known GWAS loci, and 22 genes that had identified by cross-cancer analyses for promising future assessment out of those. And so that's where the work is right now. What happens with this kind of work is we need now the experimental biologists to come in and figure out what the function is. Very hard. That's easier said than done. But that's what these kinds of studies should lead to. And I did work with Simon Gaither at the time, who is doing that kind of work. So that grant funded the work I showed you, plus his lab, to look at functionality of some of the genetic association that we were finding. So finally, this takes me to more contemporarily where I am right now, and that's gene expression and looking at subtypes and prognostic signatures. So the cancer subtypes and molecular signature characterization, to me, is a paradigm shift, leads to a paradigm shift in our understanding of cancer heterogeneity. It provides the rationale for the identification of prevention and treatment of cancers, and it supports that, again, that ovarian cancer is a heterogeneous disease that which we know about, but it's more heterogeneous even within the histotypes that I'll be showing you. And we can see differences in the molecular signatures in terms of age of onset, in terms of survival. And so within high-grade serous ovarian cancer, we can see a bit of heterogeneity that's defined by these molecular signatures. So just by focusing in, and I've been around long enough where we didn't really realize how much we needed to look at histotype in my field. And only because of consortia like the OCAC could we ever assemble enough data to consider that. Any one study that either when I was conducting or anybody else, really, when you start looking at the heterogeneity of this disease, just didn't have this power to really look at histotype as a separate entity. But as we were able to do that and classify ovarian cancers that has now been revised, and doing large-scale genomic analysis, we have been able to identify novel molecular subtypes that are associated with distinct biology and clinical outcome, and has also been used with high-grade serous ovarian cancer, again, the most common histotype that's really related to BRCA1 and 2, certainly in that DNA damage and repair space in terms of genetics. Others have realized that there are some treatments that work in this group that where DNA damage repair underlies the response to treatment. So in the high-grade serous, generally what's involved with BRCA1 and 2 and DNA repair deficiencies. So the enzyme poly-ADP ribose polymerase, or PARP, has been found to play an important role in DNA damage repair and genome stability. And PARP inhibitors inhibit DNA repair pathways and cause apoptosis of cancer cells especially in homologous recombination deficient cells in HRD. And now HRD is becoming one of the hallmarks of when people are diagnosed with ovarian cancer, and especially high-grade serous ovarian cancer, to identify whether they are HRD positive. And that will inform treatment avenues. So that it takes many years. It's this, seeing how the picture's forming in terms of really getting into something that would impact patients. So out of the OCAC consortium arose ATA, which is the Ovarian Tumor Tissue Consortium, focused on FFPE tissue because this is really what we can assemble on a large scale. And in this case, there were 3,700 or more women with high-grade serous that were pooled from multiple studies, and RNA was processed using nanostring technology. Expression levels of 276 genes were found to be associated with ovarian cancer survival, and the best performing prognostic signature included 101 genes enriched in pathways. Some novel genes were also found using this approach, but finally, from all this work, the ATA spot gene expression signature was identified or defined, and that might improve in the stratification in clinical trials by identifying those patients who are least likely to achieve a 5-year survival, a good 5-year survival. So the spot score is what we're referring to it now, does distinguish ovarian cancer patients with high versus low probability of survival. And the top quintile, just to illustrate it, has a median survival of about 9.5 years compared to only 2.3 years in the bottom quintile. So that's not in popular use yet, but that I'm aware of, but maybe some of you are, I don't know whether you've considered this, but I think my group's interested in doing more work in that area. So we'll see. But a lot of the work I'm presenting right now is from 2 studies that I conducted over many years. One is the North Carolina Ovarian Cancer Study, and the other one is the one that I mentioned, the African American Cancer Epidemiology Study, or ACEs. And at least I guess in some ways I had the foresight to collect all the paraffin-embedded tumor tissues that I could collect. We had blood in most people. We had a lot of other pathological data and epidemiologic data all matched together, which is a very rich epidemiologic data set. So recently, Lindsay Collin, who was a postdoc at the time and just joined the faculty in my department at Emory, published an abstract trying to validate that spot score. Instead of using the nanostring, what she looked at was our RNA-seq data that we had generated from the paraffin-embedded tumor tissue, and to then calculate the spot score on both white and Black women coming from those 2 studies I mentioned, the NCOCS, North Carolina Ovarian Cancer Study, and ACEs. And what she sees here, and it's just, I won't have time to go into it, is she sees a fairly reasonable relationship between the 1st and 5th quintile of the score in whites, with as high of a magnitude as 2.21 in the 5th. But in Black women, it is very attenuated. And this is using the same methods. We don't see a strong relationship, some suggestion of 1, but compared to 2, it's closer to 1 in the 5th quartile. And so the other thing that she noted was that the Black high-grade serous ovarian cancer cases, there were 42% in the 1st quartile compared to the 15th quartile with 53%. But in contrast, among the, that was the Black cases, but compared to the white cases of 58% versus 47%. So there are differences that we're observing. Can't say that I completely understand all that. So I know I have a little bit of time. So I'm going to go into what are the gene expression subtypes of high-grade serous ovarian cancer. It's there are 4 that seem to hold up in our data as well as others. The mesenchymal, immunoreactive, differential, and proliferative. These are all, this is again, the heterogeneity of high-grade serous ovarian cancers. And they represent different survival curves. Their survival is different genes in these are molecular signatures that we use to using k-means correlations. And what we did in this slide is we compared the Black cases to white cases, and from my two studies that I mentioned, as well as the TCGA data that was obtained from fresh frozen. So, when you're going from paraffin to fresh frozen, we're not sure what we'll see, but we did see a lot of concordance in different populations, including the whites and TCGA, Mayo Clinic's data, which was fresh frozen, and a Japanese cohort, and the whites. So basically, we saw these intrinsic subtypes of high-grade serous were similar across all these populations. We did not see differences. And what we did see is some differences in the proportion within each of these clusters, where of these subtypes of high-grade serous, where we saw that the immunoreactive subtype was more common, and the differentiated subtype was less common in the Black cases compared to the white cases in the Japanese populations. So there were some differences. But we also looked at the survival curves across all these populations, and we didn't see differences in survival. We saw differences in the proportion of these subtypes, but not in the differences in the survival. So both Black and white women had 20% better survival if they had the immunoreactive subtype, and there were no differences in the survival of Black and white women with the differentiated subtype, as an example. So the questions we were trying to answer from this work was, are self-identified Black women more likely to have a gene expression subtype that is known to have worse survival, and used methods that rely on the patterns seen in the self-identified white women. And basically, gene expression subtypes, as I mentioned, were similar. There was a similar pattern across all those populations, and the results from the paraffin-embedded tumor tissue compared to fresh frozen were also similar, which was interesting to note. Then another question that we were trying to address is whether Black women had different proportions of gene expression subtypes compared to white. And we did see some differences in the immunoreactive one compared to the white and the Japanese one populations. And both Black and white women with the immunoreactive subtype had better survival. So again, the proportions are different, but the survival was not different. And only 7% of the Black women were found to have the differentiated subtype compared to a much higher percentage, 22 to 31% in the other population. So that we did see differences in the proportions. And the prevalence of gene expression subtypes varied then in Black compared to the other groups, but the differences did not appear to explain the survival differences. So finally, others in our group as well have looked at these intrinsic subtypes in epithelial and high-grade serous ovarian cancer, and do see differences in survival with the mesenchymal subtype being worse, leading into better survival in the immunosuppressive and the differentiated subtypes. There is an association with residual disease in the mesenchymal subtype, which has the poorest survival, has been reported. And then in our group, we looked a little bit further using the ADA cases. And I should have mentioned that maintenance therapy might be more beneficial in progression for two of the subtypes. But in our group, we did look at some of the other data that we had assembled. As I said, we had a lot of survey data. So we saw improved survival in with hormone replacement therapy in the mesenchymal subtype. We saw some differences in mortality with a high BMI in two of the subtypes, mesenchymal and immunoreactive, and increased mortality with formal alcohol use in the proliferative and the differentiated subtypes. So whether these things hold up over time, this is kind of where we venture in to try to understand either the etiology or prognosis in these subtypes and what's driving them. And this work continues. And then the last thing I'll mention is I have a study that we're doing, a pilot study in 3 sites. At Emory with Susan Monisset, at Moffitt Cancer Center and at the Huntsman Cancer Institute that we call Ovarian Health and Lifestyle Study. And I won't go into the details of that, except that we have submitted a grant where we do propose to use the AUDA spot to see how that works in our new population that we're collecting from clinics rather than population-based studies. So I won't go into it any further, but I want to at least acknowledge all the people at Emory, the ACES team, and my gene expression team that contributed to all this work. Shout out to Jen Doherty for sure, with a lot of the gene expression work that I presented, and and Melissa, who's been part of our ACES team, and Lindsay Collin, who is a new epidemiologist coming up that I think you'll be hearing from. So lots of grants from NIH contributed to this work, and here are most of our team members. So, thank you.
Thank you very much, Dr. Schildkraut, for sharing your important work on the molecular and genetic epidemiology of breast and ovarian cancers. We have a few minutes for a couple of questions from the audience. Yes, Dr. Kapp.
Thank you for an excellent presentation. When the subgroups of the high risk, did you look at the percentage of
We have that data, and actually it's just coming out in publication. Yeah, the HRD, you mean? Yes, we have that. We have done just a descriptive paper right now. About half of them have it, if I'm remembering correctly. And
if you control for that, do the 4 subgroups still
we
hold?
will. Yeah, we just got that done. That's in progress. We have a lot of balls in the air, so to speak. Yeah, so we plan to look at that and look at the survival curves with HRD.
Thank you.
Yeah, but nothing yet, soon, hopefully.
Well, please join me again in thanking Dr. Schildkraut for her excellent work and presentation.